Hello everyone, today it's my pleasure to introduce our latest research. After long-term continuous cropping, tobacco can alleviate replant problem by recruiting special microbes to degrade autotoxins and maintain plant and soil health. Continuous cropping often results in severe replant problems across various crops due to the autotoxins accumulation, soil acidification, pathogens proliferation, and microbial dysfunction. We found that long-term continuous cropping can alleviate the tobacco replant problem. This mission occurs through the enrichment of autotoxin-degrading microbes, and the transformative impact is evident with 10% of long-term continuous cropping to short-term continuous cropping. So, our investigation has pinpointed specific autotoxin-degrading bacteria, which exhibit the capacity to alleviate tobacco replant problem in short-term continuous cropping. So, their autotoxin-degrading mechanism using axenic culture and soil samples was also conducted via comprehensive analysis of microbiome approach. This research sheds light on the potential of long-term continuous cropping as a strategic approach for sustainable agriculture, addressing replant problems and promoting the health of cropping systems. Highlights. First, long-term continuous cropping alleviates the tobacco replant problem, and this alleviation can be transferred to soil of short-term continuous cropping systems. Second, tobacco recruits autotoxin-degrading bacteria that mitigate the replant problem. Third, inoculation with autotoxin-degrading bacteria helps to alleviate the tobacco replant problem. We collected soil samples from tobacco fields in Yunnan Province, China. We found that long-term continuous cropping soil had significantly lower concentrations of autotoxins and reduced soil acidification compared to short-term continuous cropping soil. Root-associated microbial community structure and composition also differed. Beneficial microbial groups were differentially abundant in long-term continuous cropping soil. These beneficial microbial groups were significantly negatively correlated with phenolic acid contents in the soil. Adding 10% long-term continuous cropping soil significantly promoted the growth of tobacco seedlings in short-term continuous cropping soil, leading to increased biomass and decreased autotoxin content. Bacterial groups were significantly differentially abundant in both long-term continuous cropping soil and short-term continuous cropping soil with 10% long-term continuous cropping soil. We isolated 2 autotoxin-degrading strains from tobacco long-term continuous cropping soil. Under laboratory conditions, both bacteria demonstrated the ability to grow in media containing phenolic acid autotoxins as soil carbon source and efficiently degrade them. Differential gene expression related to stress resistance and autotoxin degradation played a crucial role in the autotoxin degradation process. When 2 autotoxin-degrading strains were added to short-term continuous cropping soil, they effectively colonized and reduced the autotoxin content in the soil. Additionally, bacterial strains promoted the growth and development of tobacco plants. These effects were strongly correlated with the upregulation of genes related to stress resistance and autotoxin degradation in the bacterial strains. Summary. In this study, soil acidification and phenolic acid accumulation are the main causes of replant problems for many crops. Long-term continuous cropping of tobacco alleviates replant problems by increasing soil pH and decreasing phenolic acids in the soil via enrichment of autotoxin-degrading bacteria. Long-term continuous cropping or monocultures of crops under certain stresses can induce soil homeostasis against replant problems, soil-borne diseases, and potentially other stresses to increase crop health.